Okay, we're gonna get started. So welcome everyone, and thank you all for joining us today. My name is Lilac, and I'm one of the organizers of this year's Quantitative Seminar Series. I will begin with a land acknowledgement. The University of Washington acknowledged the coastal people of this land, the land which touches the shared water of all bands and tribes in the Squamish, Tulalip, and Makosha nations. We also acknowledge that we live and work on the ancestral land of the Duwamish people, the first people of Seattle. Now let's introduce our speaker. Dr. Matthew Helmus is an associate professor of biology at Temple University, where he co-leads the Integrative Ecology Lab. Trained as a quantitative ecologist at the University of Washington, Madison. No, University of Wisconsin, Madison. I'm so sorry. He completed the postdoc fellowship at the University of Chicago, the Chinese Academy of Science, and correct me if I'm wrong with my Dutches. Bayer Universiteit Amsterdam before joining Temple's faculty. Dr. Hammers focused on how ecology and the evolutionary process interact naturally to generate biodiversity and how humans disrupt those processes. He addresses questions within both invasion biology and island biogeography using big data statistics, mathematically modeling, and field research. Today, he will talk about his work developing open source tools and interactive web apps that helps agriculture agency and farmers track and manage emerging pests. Dr. Hamas will take question after the talk. Online folks, feel free to drop question in the Q&A box as we go. Thanks everyone for coming, and big thank you to our speaker. And with that, it's my pleasure to turn it over to Dr. Hamas.
Great, thank you, Lilac. You can hear me just fine, right? Great. Okay, so today I'm going to talk to you about some of the work that my lab has been doing on spotted lanternfly and the open source data science workflows that we've been developing. And so I'm going to start off by just talking about the invasion process. And so if we have a species, that species has to pass through and get transported to different locations. It then has to establish in a particular area, and then it causes impact. And if it passes through those different, let's say, filters, then it becomes an invasive species. And that is termed invasion process theory. That's the way that a lot of invasion ecologists and biologists think about the invasion process. So if transport, establishment, and impact potentials then are high, then there's a severe risk of the species becoming an invasive species. And so we're going to use this sort of framework in order to understand this species here. So this is a spotted lanternfly. I'll use it in my slides, SLF, and I'll also spell out the name. It was first found in 2014, really close to Philadelphia. It only has one generation per year, and this is essentially what it looks like. Its eggs kind of look like this mud here. They then hatch and they molt into these little black instars and eventually into a red instar. And so these are the red instars. They then eventually molt into these flying adults, and here they're mating. And then they have gravid females is what they look like here. And then they lay eggs and they start that process over and over again. This pest species really likes a tree of heaven, Alanthus altissima. It's a globally invasive tree. It's found all across the globe. This is sort of what it looks like. The leaflets have sort of these little thumbs at the bottom of them. That's how you can sort of tell whether or not it's a walnut or other species that kind of look like this. And then I looked at INAT, and there's one just, right there on University of Wisconsin or University of Washington campus, found in Burke-Gilman, right next to the UW greenhouse. And so, they're in Seattle as well. They're all over the place. So when spotted lanternfly invades an area, especially an area that has a lot of tree of heaven, you have these massive outbreaks. And so this is sort of one of the first pictures that started coming out of the massive outbreaks that were happening here in the Philadelphia region. But they're happening all over the place. So this is some places in Toledo. They climb buildings, they climb poles. There are a lot of examples where people have to use leaf blowers to sort of blow them off, all these dead lanternfly everywhere. And so they really, really are just everywhere when they outbreak, and they climb on people, even people that don't even know that they're there. But the major impact of spotted lanternfly has to do with their feeding, and a lot of it has to do with their honeydew. And so this is a plant hopper, and plant hoppers will stick their rostrum into the phloem of a plant, suck up that sap, and then they have this sticky, sugary waste product that they eject then, and then that will mold a lot. And so not only does its waste product mold really easily, but it also feeds on a lot of other species of plants besides tree of heaven. And one of the other plant species that it loves is grape. And so here are some pictures from some vineyards in Pennsylvania. You can see all the eggs that are laid here on this post, all those lanternfly just covering, these grapevines here. And so this is a few quotes from back in 2023 of spotted lanternfly for some of the wine grape grower collaborators and stakeholders that I work with. So the population of spotted lanternfly in the vineyard has been increasing every year since 2017. Spotted lanternfly remains one of the most limiting ecological constraints to sustainability of our winery. And then we've lost about 20% of our vines of spotted lanternfly. And so this is having a really big impact on the grape growers in invaded regions. And so that's just a general overview of this pest species. And we're going to use this invasion process theory to go through how my lab has been working on transport, establishment, and impact. And now I say my lab, but really this is a collaborative research project. And so I'm going to be presenting research that's done by this team here. So Dr. Jocelyn Bem, she does a lot of the field biology work. I do a lot of data science work, and then Benjamin Seibel does a lot of the mathematical modeling. And then just to be clear, so we're at Temple University, and this is in Philadelphia, and so we've been dealing with this pest, right from the beginning, and so we have a lot of institutional knowledge about this pest. But really, it's not really us that do the work, it's everybody else, graduate students, postdocs, and undergraduates. And so I'll try and put the picture of the person up in the top right-hand part of the screen of whose work I'm talking about. And one of the things that we've learned by working with, big groups of undergrads and graduate students is that a lot of them really want to make a difference and really want to think about how they communicate their research to stakeholders. And so my lab has been thinking a lot about that. And what we've sort of thought about is how we use our coding workflows for broader impact. And so one of the first ways that we thought about this was we do a lot of work where we're dealing with stakeholders. And so we came up with a framework to generate stakeholder reports using the exact same code that you use when you're writing your dissertation or making your publication analyses. And if you think about the stakeholder while you're writing your paper and you're using that coding workflow, it's pretty easy then to just use, our Markdown or other types of, languages to then produce a report that then you can give to your stakeholders really easily. And now this idea of thinking about developing stakeholder reports in parallel to doing the analyses for publication has led us now into working a lot on building apps. And so the apps that I'm going to show you today are all on spotted lanternfly. We have a lot of different apps for all the different research projects that we're doing. And so when we think about a research project, we always think about what publication we're going to get and then what the stakeholders are going to get, and that typically is an app. And so we've kind of come up with this workflow that we think about when we're doing open data science for invasive species management. And you'll see these themes when I start to talk about the individual examples for spotted lanternfly, where we focus a lot on data synthesis, and then our modeling of model forecasts or risk assessments, and then we have some type of app release. And you have software and that cyber infrastructure that links this up. But then as we're developing these workflows, then we are thinking a lot about the stakeholders. And so in our case, we're dealing with researcher stakeholders, growers, and also agencies. And those researchers and growers and agencies all either use our products or contribute to those products by contributing data or insights, et cetera. And so that's just in a nutshell sort of how my lab and how the people who I work with in my lab, how we sort of approach these different questions of spotted lanternfly transport, establishment, and impact. So first, we're going to go ahead and talk about this question of where is spotted lanternfly spreading? We're going to talk about transport potential. So first, I'm going to give you an example of a collaborative data synthesis that we've done that really forms the basis for a lot of our research. So I showed you all those pictures of spotted lanternfly. It's a species of really high concern. And so there's lots of state and federal agencies, primarily USDA, that are surveying for it. Now, every agency, especially at the beginning, but every agency has their own way of surveying sites for Lanternfly, often collecting data very differently through time. And so this causes a huge headache when you're trying to do, a large data science project, trying to understand where spotted lanternfly is at. So working with a postdoc, Seba Devona, we came up with a framework of how you compile big data on invasive species spread. Now, I'm going to give the publications or sort of list of some of these are in press, some of these are submitted, etc. I'll give those links down here. But then, if you're really interested in a particular slide, then I've got this QR code here that you can just point your phone at and grab it. And then, of course, at the end, if you still have questions about things, I'm happy to answer questions. And then, if you still didn't get it or know where to look, if you're interested in a particular insight, just email me and I'll send you the publication or send you the link to the app. So I'm going to go through this framework here. So what we do first in this framework is we collaborate with state agencies. We talk directly with their surveyors, and we collect their data through these planned surveys. And typically these surveyors have their own ideas of where they're going to go and sample. And then we work with them to get those data from them so that then we can put them into our data processing pipeline. And so we then are processing our data from different sources. We're doing a lot of tidying of them, checking for errors. This actually takes a lot. We have huge code databases that takes data from all these different agencies and puts them all together and combines them into one data set. And then importantly, because a lot of these agencies are surveying on private property, we then anonymize the data, remove any sort of personal information from the data, and then we also round the coordinates up. And so we provide then open access data, not of the individual GPS points, but as a 1 km grid across the United States. And so then we provide all these data, mapping functions and summaries as an R package. The LIDA mapper R package is what we call it. And so right now, let's see, I just talked to my team today. We have over 1 million spotted lanternfly records now. This is from 2023, but we have about, 1 million spotted lanternfly presence, absence, and abundance records and survey records all across the United States. And so we can use those data then to try and understand spotted lanternfly spread. And then we've built apps then, such as this one here, a simple app that my team has built, sort of visualizing these spread data so that stakeholders and also the public can kind of see, where lanternfly is spread to next. So there's two different components to spread. There's diffusive spread and jump dispersal spread. And we'll go through both of those and how we're looking at that. So diffusive spread with invasive species, that's just diffusion. We're sort of thinking about how it's spreading out, let's say naturally, through a landscape. And there's a few issues you have to deal with when you're trying to understand diffusive spread. This work was all done by my postdoc, Joseph Keller, who's currently with me. So if you think about it, if you're dealing with point locations of sampling, you have to think about how you discretize or how you grid out those resolutions in order to get an invaded area. And so diffusive spread then is expected to be faster when surveys are aggregated to coarser resolution. So say you have a fine resolution here where you add up, all these pink squares and say, well, this is the invaded range or this is the invaded area versus this coarse resolution. And so we see that, of course. So for spotted lanternfly, here I plotted the total area that's invaded. Here I'm just using grids, so a fine resolution grid versus a large resolution grid. And here I've just got the years of the invasion process here through time. And as you can see, I'm just fitting a very simple line through that. That line gets steeper and steeper as you get larger grids, which makes sense. You're saying, more and more increasingly that area is invaded, of that area. So that makes sense. We'll deal with how we optimize this spatial scaling in a second. So the second thing that we need to think about when we're dealing with trying to estimate sort of a diffusive spread rate is that these spread rates are expected from theory to vary over time. So a lot of species show this pattern where you have, a lag phase in how fast that species is spreading. Eventually you get a really rapid spread once it really takes off. And eventually you hit some type of saturation or sort of a flatlining. So you expect, at least from theory, that you have some type of nonlinear relationship. And this is what we see with lanternfly as well, even though this is a continuing invasion. Regardless of if you look at, a 2 km grid or a 5 km grid, sort of these, curvilinear lines here, these red lines here are fitting the data a little bit better than these blue lines, for example. And so now the question is, if we're trying to estimate spread rate, we have to think about it in a nonlinear way. And secondly, we have to deal with this issue that it kind of depends on how large the scale is that you're looking at, and that really determines your spread rate. And so what we did was we developed a methodology, and I'm just going to sort of hand wave here, and you can take a look at the paper, and I'm happy to talk more about it. where we try to identify the optimal spread. And the way that we're trying to identify this optimal spread is by thinking about it from the stakeholder perspective. So the stakeholder wants to know the spread rate because they want to find as many invasive sites as possible. They want to find those sites where it is invaded, and they're not too worried necessarily that maybe they said that this one site was invaded, but it wasn't. They want to make sure that they find all of them. And so we first decided that these grids maybe don't make sense. And instead we went to a method called alpha convex holes. That's sort of what this pink shape is here. And we compared the fits of alpha convex holes to grids using cross-validation and found that they fit a little bit better. So what we did then was we fit alpha convex holes at multiple scales. And then we used cross-validation with this metric called the F beta metric. And it balances precision and recall. And this is really important. There's lots of different goodness of fit metrics out there. For those of you who do this type of work or for any type of model fitting, there's lots of different metrics out there. And you have to think about what these different metrics are telling us. And we found that this F beta metric, at least for this particular instance, is really useful. And the reason it is that it allows us then to think about precision and recall. So if you have a goodness of fit metric that has very high precision, it avoids those false alarms. So calling an uninvaded site invaded. But at the same time, if you have high recall, it avoids those misses. So it avoids those misses where you're failing to detect invasive sites. And that's exactly what the surveyors don't want. They don't want to miss out on invaded sites. And so using this cross-validation technique and looking at different betas, what we found was that a 35 km scale with a beta equal to 2, which basically weights recall twice as much precision, that was the best at including all those invaded sites, even at the cost, of course, of some false positives, for example. So we have now a methodology that I haven't gone through very much, but for those of you who are following along, I've said that then allows us to then get an optimal scale to estimate spread rate at. And so by doing that, what we've estimated is that for Lanternfly, at least back in 2017, that was the peak diffusive spread rate at about 20 km per year. And now, currently in the data that we have, it's only about 4 km per year. And so it's about an 80% decrease. And so there's been a large drop in spread rate. And so now what we're doing is we're trying to figure out why that is. A big question is whether or not this slowdown is due to management, because there has been a massive amount of management, in this landscape with spotted lanternfly. And so that might be one of the reasons why diffusive spread rate has slowed. So now I'm going to talk to you about the other component of spread, jump dispersal spread. So jump dispersal spread, this is that long distance transport events that occurs whenever people transport, a species really long distances. And so here I'm going to show you some work by Nadège Villard, and she did work looking at this jump dispersal spread. Here I've got just all the point locations of spotted lanternfly when it was first found in the United States in Berks County. So here's Philadelphia here. And as you can see, it expanded and expanded. That's sort of that invaded area here until 2017, where we started identifying jump dispersal events. And so we developed a methodology, and it's an R package that you can use that looks at the spatiotemporal distributions of the species, then tries to optimize the sort of gap distance among disjoint distributions, and then comes up with identifying the number of counts and where different jump events have occurred in a landscape. And what we do is we look at sort of, whether or not jumps are past a particular optimized gap size in a distribution. And then because this distribution of lanternfly spread and most species spread isn't fully uniform like a diffusion like you think of, it's heterogeneous. We take that distribution and we essentially subdivide it and divide it into different sectors and recalculate those jumps and then do optimization that way. Again, this sort of, I'm hand waving how to do it, but we have a whole R package out there that allows you to do that. And so we've identified 2 then in 2017 of these 2 jump dispersal events. This was just like a single site, an isolated site, and this was like a cluster of sites that were found, and that's the difference here. This is 2018, we get a pretty major jump to Virginia here. This year, we're getting now Pittsburgh, major jump in Pittsburgh. Now I've zoomed out, right? So now we've got an Ohio location right here on the border of West Virginia, Ohio. We've got that Virginia location. Now we've got New York and Cornell University. That was the first one. Increase in area with all those different jump distance, et cetera. And now we've actually had them in Chicago, in Atlanta, et cetera, and those other sorts of places. And so what we find by looking at this is that while the diffusive spread is slowing down, like I showed you, that jump dispersal is accelerating. And so here we partition through time the amount of invaded area, this is the invasion radius here, and how that's increased through time. And if you just look at diffusion, that's more or less sort of like a linear increase. That rate is more or less, slowing down like we talked about. But if you include that jump plus diffusion, that's increasing rapidly. And so also jump number and distance has also increased through time. So remember, there was no jumps between 2014 and 2016. So we're looking at the jumps in 2017 through 2022. And those number of jumps, has been increasing through time here. And we looked at both the spatial rarefication versus not. And then the distance of each jump from that core is also increased. So here's our histogram, our count of jump locations on the y-axis, and this is distance from the core. And these are, in 2021, 2022, those jumps, and all that, 2017 and 2018, are all over here. And so we're getting faster, more jumps, and then longer distance jumps that we're getting, and that's rapidly accelerating spread across the United States. So we have then to think about, okay, well, these jump dispersal events are unique locations where a long distance transportation event has occurred. And so what are the property types here? And so this is actually one of the property types. This is a rail station. And so when we think about jump dispersal events or invasions in general, we have to understand what invasion pathways are of particular species. And for lanternfly, it's the life stage of eggs that are transported the longest. Like I showed before, these eggs kind of look like mud, like a little spider mug, and they lay their eggs on almost anything. They seem to really like rusty metal and stone. And sure, they also lay their eggs on trees, but they'll lay their eggs sort of anywhere. And so it was likely landscaping rock with eggs that were imported from China to the US circa about 2012 that caused that first invasion in Berks County, Pennsylvania. And like I said, eggs are normally laid on stone, wood, metal, train cars a lot, pallets, and also containers like shipping containers too. And so these particular properties then that are pathway properties, where these convergence of vehicles and goods and people, where they facilitate jump dispersal, at least for lanternfly, these are these invasive species transport hubs. And so if we have a data set, which we've built, of different property types, we can start to say, well, what property types are most likely then to be associated with a jump dispersal event? So we have then our method that I talked to you about, where we have now quantified at least through 2022, and now 2023 and 2024, we have about 180 jump dispersal events all across the United States. And now we can look and see, well, what property were these different jump dispersal events associated with? So this is some preliminary results that my postdoc is working on for publication. And what he found was that by looking at these point of interest databases, these property type databases, he found that there are 8 types of transport hubs that they're associated with. Don't worry too much about what I'm showing here. Look at the stars here, but all of these are important. But basically, we're doing a null model approach. We're doing 2 different types of null models. And that red star is outside, the 95% confidence intervals of these different points. So shipping distribution centers, truck rentals, stadiums, flea markets, landscaping businesses, truck stops, amusement parks, and those railroad stations as well. And what we found just in general is that if a site has more rail in it, then it's strongly associated with a jump dispersal event, like what I showed you before. Where if we look at primary roads and secondary roads, those places that have lots of roads aren't really associated with it, but anywhere that has a high density of rail is strongly associated with it. And that's sort of what I'm plotting here on the x-axis, the sort of the density of the amount of rail that's in a particular cell or grid cell that we're looking at. And so what we've done with all of this work is then start to build applications for organizations. A lot of times it's state agencies, like agricultural agencies, then to map out these potential transport hubs and then go and survey them. And so we've been able to identify particular locations in Pittsburgh and other places that surveyors have gone to, and they've found like a new population of Lanternfly that was never discovered there before. So we have these interactive maps out. We have about 46 of these apps out across 25 different states, and we're working with a lot of them. We have one for Washington already. And of course, if you're out there and you're working in a group or something, contact me and we'll try and develop an app for you. So that in a nutshell is sort of, what we've been looking at with the transport stage of the invasion process. And so now we're going to talk about the establishment phase stage. And we're going to focus on this question of how does climate affect establishment? We're going to be doing that both with statistical niche models and also first principle population dynamic models. We'll talk about the statistical niche model work first with one of my master's students who just graduated. So we developed an open science workflow that assessed the shifting risk of invasion with climate change. And this link here will actually take you to an R package and research compendium that looks at all this. I'm not going to really go through the entire thing here, but instead I'm going to sort of give you an overview of what it is that we're doing. And so what we've done is we've developed this R package and then a whole publication as well, where it'll look at the shifting establishment potential of Lanternfly with climate change and correlating that with important viticultural regions for different nations, for any geopolitical unit. So I'm just going to focus on the United States here and talk to you about what I mean here with these risk graphs here and then these risk maps here that this package will produce. So we're building statistical niche models. So we're taking all that presence data that we got from the Lycorma package that we put together, and we're building here, Sam just used Maxent as a standard model. And we're using spatial data from both the native range and the invaded ranges. And what we decided to do was we contrasted two different projections of spotted lanternfly establishment potential using those occurrence records, modeled on covariates, temperature and precipitation covariates, that are predicted to shift with climate change. So first we did what is basically done by anyone who does, niche modeling. And I'm sure, many of you have done this, or certainly all of you have probably read papers on this, where we use all the data in one model. And that's kind of what's typically done. And so here's, our data in the United States, and here's the data in that, the Asian range here. So we can put all those data and build one statistical model. Then what we also did was we regionalized the data, because each one of these regionalizations corresponds to distinct evolutionary trajectories or histories of the species, different populations. So we have a regional data model where it's just a North American invaded range. We have one here where it's just that native range, so it's native to China here. And then we have one with the invaded Asian range, which is in South Korea and also Japan. Those are also areas. And we ended up ensembling those regional models. So now we got two different projections. We got a global data model projection, which is similar to what's typically done, and then an ensemble of these regional model projections. And the reason this is important to regionalize your data and look at differences in your data is that for Lanternfly especially, they are regional specific climatic responses. And so that means there's geographic variation in its ecological niche. And here's what I mean by that. So what I have here that you can think about on the y-axis here, this is sort of establishment potential here. And then on the x-axis, this is that covariate, range and mean diurnal range. And you see here, this black line is that global data model, and that green line is the native model. You can see it's very, very different profile is what we're getting. Same with annual precipitation, same with precipitation seasonality. You got a very, very differences. Now, the only one that sort of is all the same is this mean temperature, the coldest quarter, and I'll come back to that in a little bit. You can see that different regions have different geographic variation in their ecological niche. And so now that we have these two different projections, we can start to ask ourselves, okay, well, what does it mean then if you have these two projections telling you the same thing or different things? And so we've interpreted it this way. So we're going to map the intersection of the global data model, global data projection, and then the regional ensemble projection. And so in areas of the globe where it shows that it's not suitable, then that would be a low risk situation. In areas where they're both saying that it's suitable, then that would be an extreme risk. In those areas where you have that ensemble projection saying that it's suitable, then we're going to say it's a high risk because at least somewhere in its range, it's really important. And then the global data model projection, this is rare, is whenever you have more of a moderate risk, is sort of what we assumed it to be. So here we've mapped these different risks across the globe. This is our present day risk. You can sort of see sort of where it is, so we expect it to be across most of the United States, most of Europe, if it spreads there, and of course, in the native range of China, et cetera. And then this is what it looks like with, we did lots of different climate change scenarios. This is an average of climate change scenarios. You can see that expansion into Northern Europe there, for example, and a little bit of a contraction here in the United States as well there. So just focusing on the United States, what we find, at least with this particular model, is that it increases U.S. establishment potential. There's a large amount of area that becomes more suitable with spotted lanternfly, but at the same time, there is particular areas that come out of suitability, as you can see here. You have that, shifting north that you sort of expect to see, and that's sort of what's expected here and here. Now, each one of these points that I have here is an important viticultural area of the United States. These are called AVAs, or American Viticultural Areas. And so we can look at then how these risks change for each one of these different points, for example. So here we have one in Arkansas, for example, and that risk is changing with climate change. And so now we can look at it in the context of a risk quadrant plot, which is done a lot in risk assessments in public health and other spheres as well. So what we have here then is the global model risk projection, that regional ensemble model risk projection, and then I've just drawn arrows as to how these different AVAs are changing. So you can see, this one here went from a high risk to a low risk regime, where a lot of these actually move from the extreme risk over to the high risk. And so what we found by looking at this is that only 5 AVAs, 5 of these American viticultural areas, increase in risk, about 53 decrease in risk with climate change, and then about 170 maintain that exact same risk. And so it seems like climate change, at least for a lot of AVAs, or a good chunk of AVAs, will decrease its risk. But to be clear, it also might make those AVAs unsuitable for growing grape. Now, if we had only built the global data model, like what's typically done, that risk is highly underestimated. And so this is what I mean by that. So this is our risk map here, and this is just the global risk projection here. Now, if we had only looked at the global risk projection, these lines here are these minimal suitability thresholds. So anything below this line means that the model says that Lanternfly can't exist there. So this entire area here would be low risk. And this entire area here would be that high risk area. And so it's missing all of these points here that you actually get when you're dealing with that regional ensemble. And so it makes sense to regionalize your data whenever you're doing these type of risk assessments with invasive species and climate change. So now I'm going to talk about some first principles modeling that we've done, again, thinking about climate. So I'm going to return to this figure where I showed that cold temperatures across these different models are the ones that were more or less consistent, that it was that cold tolerance. This is, mean temperature, the coldest quarter, because the colder that it gets, the more likely that the eggs, when they're overwintering, will freeze and die. And so there's a really strong selection pressure for that across these different regions. And that's because spotted lanternfly is poikilotherm, right? These are insects. So these animals whose internal temperatures vary considerably with these ambient temperatures. And so when we're trying to understand population dynamics of a poikilotherm, you have to think about that from the perspective of these temperature fluctuations. And so population growth is tightly related to temperature. These are sort of these thermal performance curves that you sort of get when you're thinking about that. And what we did was we developed a system of partial differential equations to model spotted lanternfly population dynamics. And this was all done with a postdoc, Stephanie Lukowitz, who's a mathematician. And we can come up then with maps that look something like this, where we got the R0, or the population growth rate, across the United States here. And anything under 1 would be, an area that's, not suitable. And I'm going to talk to you about how we made these models. So we developed and again, I'm just going to be sort of hand wavy here. We developed a moving mesh numerical framework. It solves for population growth rate and population dynamic models. For those of you who work on population dynamic models, take a look at this paper, see if it's a method of optimization that you could use. But basically what we did for the purposes of this talk was that we took locations across the United States. We then got their average seasonal temperature profiles, right? Here in Detroit, and here in Napa, et cetera. And then we ran those through our models to calculate R0, is what we've done. Now, R0 itself is a combination of a whole bunch of different temperature-dependent vital rates, mortality, development, fecundity, et cetera. And so there's lots of different vital rate functions that go into this, and it's pretty complicated. So here, for example, are all the different vital rate functions. I'm not going to go through with how we did this, but I'm going to talk about how we parameterized just one of them. So we collaborated with a lot of entomologists. We worked with them directly, and we're able to get their raw data and synthesize their raw data, where we're looking then at temperature-dependent vital rates, where these scientists have either measured vital rates out in the field or measured vital rates in the lab. And we got all our data together. I'm just going to look at one, vital rate function. This is a slightly older function that we looked at. It's a little more complex now. But this is for non-diapazing eggs here. And so mortality rate should increase here once it hits, about, a 0 or so is what it is. And anything after that, we don't really know anything about. And we're going to say it's 0. These are the actual data. When this paper was published in 2022, there were only 4 data points that we could extract from our colleagues on how decreasing temperature then increases egg mortality rate. Just 4 data points. Now, this is 4 data points, right? And so various vital rate functions, like what we need to put into our system of equations for the population dynamic model, various vital rate functions can fit equally well. It's 4 data points. You can basically fit anything. A single breakpoint model, exponential, double breakpoint, they all fit exactly the same. But they don't fit the spatial data exactly the same. And that's because this area here, where that minimum egg temperature, where you start to get increases in mortality, is incredibly important. And so small variation in how we fit in those different models causes major differences across space here. So here, for example, is the single breakpoint, the exponential and double breakpoint that it doesn't match any of the current state at all. So you have to really think about your data sets when you're doing these population dynamic models and what assumptions you're making and how little errors can have major biases in spatial distributions. And so we've also built then an app to sort of look at all this as well. We've not just looked at climate change with the mean or average seasonality, but also variable seasonality. So we know with climate change, we're expecting to see, more extreme temperatures, both, higher highs and also, lower lows as well. We want to know how this temperature variability affected population growth rate. So we simulated population growth rates, spotted lanternfly, across the United States, and compared both that average or mean versus the variable temperatures. And I'm just going to talk about 2 of these locations, Napa and Yosemite, because they're really close together spatially. So I'm going to show you a few figures and ask this question of how does temperature variability affect growth? This is simulated growth rate. So I've got my population dynamics model. I've got 1,000 simulations that I'm running through it. And then I'm going to increasingly make temperature variability higher and larger, daily temperature fluctuations, higher and higher on this y-axis. This is the R0 value. I'm going to plot R0 values here, where it's from just the mean seasonality. So there's no variation at all. And then this is the equilibrium growth rate. So below this here would be a population decline. So if variability causes higher growth, the values that I'm going to plot are going to be up here. Lower growth would be here, and then a decline would be there. This is Yosemite. So we're increasing variability, as you can see through here. And pretty soon, once you get to, moderate levels of variability, the R0 value, which we have over here, that R0 value crashes, right? And so in Yosemite, at least, if all the other aspects of lanternfly survival are met, just a little bit of climatic variability could cause those populations to crash. This is Napa. So Napa is in this sweet spot where the variability, now some variability can cause, wide changes. And so some years, just depending on, how warm it was during the winter or how cool it was during the summer or just, random changes can cause massive outbreaks. So 2 times higher growth is what we get up here in Napa. And it takes a lot higher variability to cause these populations to crash. So there are particular locations across the United States that are very much buffered from this variability. And this variability then could be something that causes massive outbreaks as well. Now, what we also found, for those of you who are following along, that if you remove the ability for lanternfly eggs to go into diapause, then we found that this population variability sort of follows a similar pattern, meaning that likely we won't see multiple generations per year, even in Napa. Likely we'll just see 1 generation per year. So these are the different counties where we're seeing this type of climatic buffering. So in areas where there's desert, we probably won't see lanternfly surviving there anyways, because it doesn't really have the precipitation profiles or the water potential profiles there. But in areas like California, all these wine growing regions here, and there's wine growing regions here too. These are places there it's likely to see outbreaks due to this temperature variability that's increasing through time with climate change. And so finally, I want to talk about impact potential here. And I want to address this question of what the risk of the spotted lanternfly is to the global wine market. And we worked on this during COVID, and we realized that the invasion process is very much like the infection process. So transport can be thought of as transmissibility of a disease. Establishment, that analogous term is called the infectivity of the disease. So how likely it is that once it is transmitted to you, then it actually establishes in your system. And then finally, impact would be virulence here. And they're completely synonymous to the invasion process. And public health researchers have a lot of public communication tools, and one of them is the Pandemic Severity Assessment Framework. I'm sure many of you have seen this, sort of flatten the curve type idea sort of came out of it. It was developed by the US CDC, and it was a framework to assess the risk of human pathogens that cause pandemics. The two main parts of it that I'm going to take from it for these next few slides is thinking about risk in terms of quadrant plots, which is what I already showed you, and also having some type of numbered risk scale. And by doing this, it allows you to communicate your results to stakeholders really well. And so what we develop then, or what we argue for, is that if we think about some species as being potentially pan-invasive species, these species can be defined as regional pests that then become globally invasive species that go out and disrupt global markets, just like a pandemic would, just like a pandemic causing pathogen would. And so if transport, establishment, and impact potentials are all aligned at the global scale for a particular species, just like kind of what you would see with a pathogen that might cause a pandemic, then there's a high risk of this species spreading and causing a pan invasion. It's causing these global market shocks or global market effects. And so we're going to address this question of, for Lanternfly, could it shock the global wine market? What's the potential? And so we took this pandemic severity assessment framework and we applied it to invasion process theory. And we came up with the pan-invasion severity assessment framework. And there's an R package and everything that's associated with it. And so starting with that pandemic risk quadrant, where public health officials, what they do is on the y-axis, they have transmissibility and effectivity combined, because typically when it's transmitted to a human, they catch it. There's not really any establishment. They just sort of catch it. And then virulence here is on that x-axis. And, for COVID-19, it would be something that looks like this. Everyone basically catches it, but that virulence is much higher in the older populations. So we took out transport potential and establishment potential and split those into two different axes. And then we thought about impact potential. And here we're just going to use scale of the points. And each one of these points here then will be a particular region on Earth. Maybe it's a state, or maybe it's a nation. I'll talk about both of those in a sense. We then look at the multivariate correlation of these three different potentials: transport, establishment, and impact. And we correlate that then to the size of the market for those particular units of analysis, for the particular nations or states that we're looking at. And that allows us to then get a correlation among market size and then invasion risk. And that then gives us a numbered risk scale that we can actually output and communicate to stakeholders then. And so I'm going to go through that, but that in a nutshell is sort of the framework. So here we've mapped the invasion risk severity of spotted lanternfly. I'm mapping establishment, transport, and impact potential just visually. So here, establishment potential is based on a slightly different niche model than what I showed before. So the more orange areas are areas that are more likely to have spotted lanternfly establish. These red lines here were where spotted lanternfly has already invaded. This was back in 2021 when we did this. And then I've just drawn these arrows showing that these states here trade the most with these following states. So these invaded states trade a whole lot with California. They trade a lot with Texas, Michigan, Illinois, et cetera, for example. And so it's showing that these areas here, like in California imports a lot of goods from this invaded area. Now we're going to look at it from the global scale. So again, we're looking at these states that are invaded and where they export their products to. They export a lot of products to Canada, some to Brazil, but you can see that major transatlantic shipping distribution network where a lot of these states ship a lot of goods to Europe, to these areas where there are a lot of vineyards. So these are all the globally important viticultural areas, all these purple dots here. So spatially, it looks like they're quite aligned. And so we can see that in our risk quadrant plots. So these are US states, these are countries here. And so the US states and the countries that had the highest transport and establishment potentials also had the highest impact potentials. And so there's a multivariate, alignment correlation about 0.41 or 0.67. So basically, if you look on this top right quadrant, if you see big circles, that means that that's the area that has the highest establishment and transfer potential. That's also the area that produces a lot of wine, if it's purple, or produces a lot of grapes, if it's a larger point size. And so you can see then that you can see visually sort of what's going on here for a stakeholder. So we take those multivariate correlations, we calculate a predicted invasion risk severity assessment, we rescale the data to be a number that ranges between 1 and 10. And then we look at that correlation between wine export market size. And so you can see, of course, those countries that export a lot, and of course, United States isn't in here because we're doing it from the perspective of these countries getting Lanternfly from the United States. Spain, France, Italy, Austria, South Africa, Germany, all these countries that are these major wine producing regions, they all have these major, they produce a lot of wine production. They have large export sizes, and they're all predicted to have really high invasion severity. And so you could look at that correlation and come up with a number where basically out of a number of 10, the risk of spotted lanternfly to shock or disrupt this global wine market is about an 8 out of 10. And so it's highly likely the species is going to keep spreading and potentially spread into Europe, and start to really affect the grape production in these vineyards. And we developed using Earth Engine in this case, a app where you can zoom in and zoom out and take a look at these different establishment, transport, and also impact potential across the globe. So just some conclusions. So first, I'm going to talk about some conclusions with spotted lanternfly, and then I'll step back and talk about some conclusions, just thinking about invasion biology from this quantitative perspective. So first, what we're seeing with lanternfly is that local spread is slowing, but that long distance spread is accelerating. And so the primary invasion front, that's expanding more slowly over time, but there's lots more new satellite populations, and they're emerging farther and farther from the core as well. And then climate change will likely alter the species range as well. So we're having these, we're looking at these projected climate scenarios. And while lanternfly will gain in suitable habitat, especially in the United States and some regions, there's a lot of important wine producing regions that are probably going to become less suitable. But again, like I said before, those wine regions might also be less suitable for growing grape itself, not just having lanternfly too. And then climatic variability can amplify those impacts to vineyards. So we have to really think about how climatic fluctuations and the interaction between the climatic fluctuations and those vital rate equations, how then those interactions can cause either there to be population decline or major outbreaks. If you just have a year where everything is just going really well for lanternfly, you could have massive increases in growth rate. And those areas, a lot of those areas are in California in the United States. And then finally, spotted lanternfly poses a pretty severe risk to the global wine market. If it continues to spread, it's going to start impacting, wine and grape production across the globe. And so there really needs to be more proactive monitoring. And so thinking about this from the perspective of, okay, someone who does quantitative invasion biology, sort of what are some take-home messages that I think are important based on, my past research and everything? Well, first, thinking about when you're doing your research, as either, professor or a student or anybody who has stakeholders, try and think about unifying your coding workflows, where you're unifying your coding workflows for publication with some type of stakeholder app or report. And by doing that, then it makes it a lot easier to have these broader impacts. Data synthesis is foundational. So you want to always synthesize your occurrence and life history data sets. You want to do that in a really, really well-curated way. And that should always be your first step. So don't short shrift that. And then make them open source as much as you can. And just know that this can take a lot of time and effort. And that is okay. So that's okay if it takes a long time. It took us, at least 2 years to sort of really get that data set together. And then when you're thinking about climatic changes, you want to model both the mean and the variability of climate. And that's because population growth, establishment potential, outbreak risk, they're driven by these complicated vital rate interactions with climate fluctuations. And ignoring that variability is going to misrepresent sort of these real world dynamics that your stakeholders will see. And then vital rate uncertainty cascades into spatial forecasts. So if you have sparse data when you're trying to build a population dynamic model or small errors, if those demographic parameters are like key to the species, they can lead to really large biases. So you really have to do a lot of sensitivity analyses with your models. And then I'm not the first one to say this, or the only one, but if we start thinking about invasion biology like epidemiology, then we essentially can adopt some of these tools and some of these effective communications, stakeholder communication tools from public health in order to improve our communication skills for policy, for example. And then finally, I would argue that you should try and build interactive applications. It's getting easier and easier with AI to build applications. Students, especially undergraduates, really like doing this. And so, if you're working on a data set and you want to mentor an undergraduate or something, a lot of, CIS graduate students or undergraduate students or other, undergraduate students have the skills to make some of these apps. And so you can incorporate them then into your research as well. Well, thank you so much for sitting through this, and I'm happy to take any questions. The apps that I talked about are at this website here, and then any publications, everything just are at my local website. Thanks so much.
That was awesome. Um, so we're now having some time for question. Online people, feel free to submit your question in Q&A box or raise your hand. I can just unmute yourself. People in classroom, I'm sorry, but you need to come over because the microphone is in my computer. Um, I think I will start with like a really general question, like how far west the spotted lanternfly has like reached. Last time I checked, it's like Indiana. It doesn't seem to move to Illinois right now, or just like want to hear some updates.
Sure, yeah. It's in Chicago. It's been in Chicago for probably at least 3 years. They just identified it last year, though. And so there's a pretty large population in Chicago. And so now that it's hit Chicago, it's going to spread west really rapidly because Chicago is a transport hub and it just ships tons of things out west and everything. As far west in the south, it's in Tennessee. I think there actually might be a population in Memphis, Tennessee, right on the Arkansas border now, but there's a pretty large population in Nashville. And then there's anecdotal evidence that it's also in Houston, in Texas as well. And then we just found out that it got to Atlanta as well in the south. And then as far north as it's gotten, it's in upstate New York. It's right on the border with Canada and Buffalo. So Canada's very worried about it. And it's gotten into Albany, et cetera, to Massachusetts, Connecticut, and those places as well.
Okay, I think I have one more question. I think you roughly touched on it, but I'm just like wondering how do you define a jump dispersal? Why not it just like a slowly dispersal, but you just didn't find the path which connects to it?
Yeah, I think that's a great question. The best way to do that, and really the only way to do that is that if you have sort of both places where you sample where there isn't lanternfly and places where you sample where there is lanternfly, right? And if you only have the places where there is lanternfly, then it's really hard to do that. But with the data set that we've got, we have a lot of samples where the lanternfly isn't found. And so we use those to try and estimate where lanternfly is or isn't. And that then allows us to interpret whether or not there's a jump dispersal event. Did that answer your question, Lilac?
Um, Sort of. I'm more worried about like because you cannot like sampling every place. It's just like, for example, you're saying like it's jumped from this place to that place, but maybe they're just like eggs in some like forest in the middle of that, but nobody's ever like looking into them.
Yeah. There is, so we try to deal with that spatial biases and where people sample, because just like what you said, you know, they might go and sample places that are easier to sample rather than the middle of a forest or something like that, right? And so we try to deal with those by doing a lot of like spatial rarefaction and other things. The other thing too is just that a lot of these jump dispersal events, it's very clear that they weren't there before or anywhere near it. The first jump dispersal event that occurred in Virginia, it was, hundreds of miles away from the main location, for example. And so for those, it's very, very clear. For some of these other jump dispersal events that, maybe are only like, 30K away or something like that, that we throw out a lot of those that get picked up when we don't have enough data to at least try and estimate that we have a good sampling in between, that we did sample in the middle of the forest, for example, those sorts of things. And it's all based on optimization, and depending on sort of what optimization you use, you get different answers. And so we try to go with the most conservative approach.
We do have a question in room.
Oh, sorry.
Oh, no, go ahead.
Hi, that was great. Thanks. I was curious what what has been the reception with your various stakeholder products, and do people tend to read the reports? Do they prefer the apps? Is it different by stakeholder? If you can speak to that a little bit.
Sure, yeah. The reports that we've done, we've actually done it. We do different things in the lab. And we've given reports to agencies based on some Lyme disease sampling that we've done, and that was pretty useful. They read those reports. We also give reports to homeowners. We went to a lot of homeowners' houses and did some biodiversity sampling here and also in the Caribbean where we also work. And homeowners were really excited because we kind of gave them a report on all the biodiversity that we found at their house, and they're like, wow, I knew we had a lot of birds and now I can see it. And so that was kind of really exciting. We don't do a lot of reports on Lanternfly. We do more of these apps. And these apps have had quite good reception. The USDA uses these apps quite a bit when they're trying to determine where they're going to go and sample next. And then the states that we work with use them as well quite a bit. Some of our apps are used more than others, I've noticed, and have been trying to figure out sort of what it is about them. And so I do have a lot of meetings with stakeholders asking them, like, do you know about this app? This is what it does. How would you like to see it differently? And so at least for those apps that we have continuous funding for, which is a lot of the transport risk apps, some of these apps are sort of just one of apps that, like, we did the best we could, we give it to people, we get their feedback, but then we don't have funding for it anymore. And so that kind of is just there, but it's still open source so someone else could take it. But for those apps that where we have continuous funding for, I do a lot of getting stakeholder feedback. This past week, even I did a lot of sort of reworking a lot of our plans for this summer based on stakeholder feedback for some of the apps that I showed you here today. Does that answer your question?
Yeah, that's great. Thanks.
We have one more question. Go ahead.
Thank you so much. I am like astounded at just the depth of your bench in terms of all of the tools and approaches that you take to integrating the research with stakeholder, like just really applied stakeholder information needs. So in Washington, a relatively new invader we're dealing with is European green crab, and we're at a stage right now where we have like a declared state of emergency for the last 2.5 years, and the state is building capacity, but we have one of the biggest gaps we face is communicating information in a way that synthesizes data from a constellation of people working in different ways and summarizing it in a way that enables decision makers to actually use the information in more than just like, we found 5 crabs here. Yeah. One of the things I'm particularly curious about through the process of working with all these stakeholders is about the conversations, particularly with not as much agencies, but in your case, like the grape growers.
Yeah.
In large part, when models and apps produce predictions that don't align with their expectations, understandings, or observations, how you negotiate that process, or have negotiated, or try to.
Yeah. Let me think. So I didn't, yeah. So that's all great question. Yeah. I didn't show you any of the work that I've done because it's all really recent, like within this last 6 months or so, really thinking about the grower perspective and what we can do for the growers. And so we have like individual based movement models of Lanternfly, because every year what happens is that the growers will spray their field. These aren't organic places. They'll spray their field, they'll kill all the nymphs, but then at the end of the year, about the time when they want to start harvesting, all the adult lanternfly will start flying in from the margins and start to then invade their vineyard. And that's where you get a lot of those pictures that I showed you, those adults just swarming them. And then they spray, they come back in, they spray, they come back in, and it's a complete mess. And so knowing the timing and the spatial and where lanternfly will invade your vineyard, that is sort of what they want, right? But that is really, really hard, right? Because you have to know the population dynamics of the species in the surrounding vegetation around a particular vineyard, right? And that's really difficult. And then on top of that, you have to then know, and at least we have this down, you have to then know like the development rates of lanternfly in that area based on temperature and that sort of thing. And so we kind of have that going well. And so right now we're pretty good with sort of the developmental rate stuff. But then that individual movement rate, that's something that we're just now approaching. And so now I'm just starting to have those conversations with stakeholders. And I am finding that like what they want is way more complicated than what we could currently provide for them. But by talking with them, it allows me to then think about what are the data that I need in order to provide that to them. And so what we've done then is basically try to find particular vineyards that have had really LiDAR mapping, of the area and other sorts of things where you can sort of start getting an idea of where all the trees are in the landscape, and then thinking about, okay, if we simulate across this landscape, where would we need to put lanternfly traps to monitor populations so that they would know when and where they'd come in and that whole process. But I feel like that whole process isn't going to be within the next year or so. It's going to take quite a few years to get to that point. And that's because those needs at the local scale are so much more complex than the needs almost at the broader scale.
Yeah.
I know that doesn't fully answer your question, but that's sort of what it made me think of.
No, that's it's awesome because yeah, the resolution spatial heterogeneity is very much a part of our challenge as well, and how to interpret a landscape. So yeah, thanks. I might hit you up later with some additional questions, but thanks for your time.
Sure, absolutely. Yeah, thanks for the question.
Thank you.
Mikey.
I know we're over time, but we're gonna address this last one question. Hopefully it's okay.
Of course, yeah.
There's an online question as like, how do you handle updating the apps used by public / stakeholders? Is it linked to your database for update in real time once processed, or is it like monthly or some other schedule?
Yeah, right now it's some other schedule. But over this next year, we're going to try and do this a little bit more systematically when we update it. One of the reasons is that we don't get regular updates of data from our stakeholders. They kind of come in at random times. Sometimes we have to chase down data and everything. And so we don't always have all the data like monthly or something. Some stakeholders will just, New York, for example, just sends us the data monthly. We just get the data and we're able to sort of update it really quickly. Because the way that we've built all of our workflows have been sort of more modular, where like every single workflow kind of does a different thing. Now we're working on trying to integrate and connect all those workflows. But because it's all code and it's all R, we can sort of do that. And so over this next year, I'm going on sabbatical. And over this next year, I'm hoping to sort of start working on how to sort of integrate this entire process. So it's much more streamlined. It kind of like you hit a button and everything runs. Like it's that sort of thing that I'd like to work on. But right now it is sort of that other schedule. But that's sort of the bane of our existence, is that we only have short funding and we only get what we can do quickly, and then how do you connect it all?
That was great. Okay, so I guess that's for today. If you have like more interesting to connect, sorry, with Dr. Helmus, feel free to reach out. And thank you again, everyone, for coming to ConSam today. And we hope to see you next week again.
Great. Yeah, thank you, Lilac. Thanks for inviting me. This has been a lot of fun.
Yeah.